Noswaith da, a croeso cynnes iawn I chi gyd. Good evening and a very, very warm welcome to you all. I'm delighted to welcome you all here this evening. And tonight we've got the privilege of gathering for another instalment of our public lecture series. Now this series allows us to reconnect with esteemed alumni who've made a significant mark on the world, and it gives us the opportunity to learn from their experiences. Dr. Tina Barsby, OBE, a distinguished alumna of Bangor University, is renowned for her scientific achievements and experience in the agricultural crops sector. She's the former director and CEO of the National Institute of Agricultural Botany and the first female chief executive in the Institute's 90-year history. In 2018, she's awarded an OBE for services to agricultural sciences and biotechnology. This evening, Dr. Barsby will explore a topic of enormous significance: the complex landscape surrounding genetically modified crops and the transformative potential of precision breeding, a subject which is at the forefront of agricultural science and biotechnology. Through her keen insights and expertise, Dr. Barsby will talk about genetically modified crops and precision-bred crops which could pave the way for a new generation of environmentally superior food production. Her insights could not be timelier. Her outstanding advocacy for scientific advancement and her commitment to serving the greater good exemplify the spirit of innovation and progress that defines our collective aspirations. As an esteemed alumna of Bangor University, her journey is marked by remarkable scientific achievements and unwavering dedication to advancing agricultural practices for the betterment of society. It's a real pleasure to welcome you here, Tina. I'm very much looking forward to the lecture. Ladies and gentlemen, Dr. Tina Barsby, OBE.
Thank you. So one of the IT people said, we were talking about the setup, and he said, it depends how you lecture. And I thought, well, I've never worked in a university, so I don't really know how I lecture, and I don't do it very often. But I just thought that this was a topic that many people are interested in, and it's something that I hope after the lecture we can have some conversation. Because I think genetic modification is a friend. And so I found it difficult for the faux part, but I do know some of the reasons why people think it's negative. Is it not working? Or have I got it the wrong way up? Yes, wrong way up. Never mind. Yes. So I think it was in 1979, it might have been in 1978, I graduated from Bangor with a BSc in agricultural botany, and then I moved to do my PhD at the University of Nottingham in potato protoplast fusion. So a protoplast is what's inside a plant cell. So the big difference that most of you will know between plants and animal cells is that plant cells have a cell wall and animal cells don't, and you can remove that cell wall and put together different species of plant, and sometimes they grow into a plant that you wouldn't have had before. So, and then I moved to Kansas State University and developed the first somatic hybrid in a crop plant, which was actually a cross between a potato and a tomato. And we got potato tops and tomato bottoms. So the plant doesn't have enough energy to actually be able to reproduce that. The light's in my eyes, I'm going to move. So it doesn't have enough energy to make potatoes and tomatoes in the same plant. So, but if you think about that, if you think about a protoplast, you're just shoving two cells together and hoping that they generate something novel. It's still used. I used it quite a lot, and I worked for a company for 18 years, and we use the same technique to move mitochondria around, so to move male sterility from species to species. So if you eat cauliflower, you may well be eating something that I worked on a long time ago. So I just wanted to start by saying that food production must undergo a transformation. We need to increase the amount of nutritious crops we grow, but also we need to reduce synthetic fertilizers and pesticides, and actually they're being reduced for us. So it's very, very difficult now for a chemical company to put a fertilizer or a new pesticide on the market. And so they only develop them for the big crops like wheat and maize and soybean, but they don't develop them for the brassicas and the lettuces and things like that. So that has to be worked on separately by farmers, by the levy body, and then they have to present to the government, this is something that works for this crop. We also need to increase the land available for biodiversity. Everyone knows that. We need to reduce greenhouse gas emissions, and we need to do this, but without just transferring all of our problems to another country. So I'm not going to talk about agronomy today, but it is a very, very key issue and a very important thing that farmers are doing. So you might have heard of regenerative agriculture. Where I live now in Cambridgeshire, you very rarely see animals, although they are now integrating livestock into the systems. I was walking my dog the other day and there was a sign saying there are sheep grazing here, and if your dog worries them, it will be shot, which was a bit of a surprise because we don't normally see animals in our area, but they are starting to do that. And these principles are the principles of regenerative agriculture. So maximising crop diversity, so having rotations, which, for many years, farmers in that area were just growing wheat and oilseed rape, wheat, barley and oilseed rape, not integrating legumes into the rotation or any other crops. The other really important thing is to provide constant soil cover, which farmers are doing now. So not leaving the land bare over winter, but putting in something, even if it's not something that they're going to harvest. So there's a lot going on in farming and in agronomy that is really good stuff. So I worked at NIAB after I left the seed company. I worked at NIAB for 15 years. And NIAB is the UK government's main technical contractor for plant variety rights. So there are one or two other places that do it for other crops, but for NIAB it was mainly the main crops, so cereals and oilseed rape specifically. And NIAB represented the UK government in international bodies that work on plant variety rights. And the reason I wanted to say that is that genetic technologies, all of these genetic technologies, can only reach the market through seed. So they have to go through a seed certification process, which takes a while. So these new varieties can be protected in the EU and in other countries following the principles set for plant variety rights. And these new varieties are tested for distinctness, uniformity and stability, as well as their performance relative to other varieties. There's a national list that's run by DEFRA and a recommended list that's run by the levy bodies. And this process can take 3, 4 years, depending on the crop. Genetically modified crops are also required to follow a lengthy and expensive process, particularly in the EU. And I know that one of the concerns that people have is that this technology has ended up in the hands of big business. Well, to me, it's pretty obvious why, because an academic institution or a small company could not afford to do the things that you need to do in order to protect a genetically modified crop. Oops. So these are sort of basic numbers, but if the world's to grow enough food for the projected global population in 2050, agricultural productivity may need to double. So if you look at maize, rice, wheat and soybeans, they're the main crops that people eat. The yield is increasing by about 1% a year, and it needs to be 2.4% to double yields by 2050. So that's just a general thing. So plant breeding and agronomic practice are driving yields up, but not quickly enough. The loss of crop protection products are pushing yield and quality down because everything you buy in a supermarket is treated with a plant protectant. So in the field, it will have been protected from disease by a pesticide or a herbicide or something. So even if you buy organic produce, I was with a farmer the other day who said, we're soon getting to the point where we're going to be able to use more chemicals on organic produce than we can on conventional. We can perhaps talk about that later. Genetic solutions are, however, available for all of this. So when we think about plant breeding, if we think about traditional breeding, you identify traits in individuals of species which you can cross. So a wheat plant with a disease resistance and a wheat plant without it, and you cross and you combine a new variety. Marker-assisted selection, I'm not going to talk about, but you can have a DNA marker which you can recognize in a laboratory, and that will identify a trait even before it can be seen. So that's a good thing. So genetic modification allows genes and proteins from any species to be moved to another, giving a new trait. So, DNA is DNA, it's your DNA, it's plant's DNA. Basically, you can transfer genes and proteins from any species to another, but there's general agreement that you don't move animal genes into plants. Mutation breeding has been around for a very, very long time. Seeds are treated with mutagens, so either a chemical mutagen or irradiation, and they promote thousands of random changes in DNA. And then the plant breeder will look at that and say, that's something I like, but then they have to cross out all of the other random changes that they've made, which they don't want. Gene editing, which we also refer to as precision breeding, you can make specific changes to DNA, creating specific mutations. And the reason you can do that is because we understand a lot about the genes that control traits in plants. So it's relatively, I say simple, but to identify which mutation you need to create a change. So the plant breeder would say, I need all the tools in the toolbox in order to address these problems. I need genetic modification, I need gene editing, I need new breeding methods, which are, again, something I'm not going to talk about, but something which is accelerating as well. And, you can look at it this way too. You need biotechnology. Crop modeling has become a very, very important part of how people grow crops. So if we think about plant breeding, so this is the science of selecting the best plants to cross with other good plants to obtain better varieties. We always used to say to the plant breeders, you cross the best with the best and hope for the best. And usually that works, you get something better than the other two. But it's slow, it's expensive, and it's sometimes difficult to manage. Sometimes you need huge plant populations to obtain the desired traits. And so a big part of the plant breeder's job will be screening for the traits that they're looking for. So a disease resistance or something else. And humans have been breeding crops for more than 10,000 years, which means they've been saying, that looks like it's got a better yield or it's got a disease resistance. And so they're selecting those and then moving them on. So if you think about wheat, which provides 20% of all calories and protein, apparently, and it's the main staple for 1.2 billion people who own less than $2 a day. So if we think about the UK wheat yield over the last 100 years has increased. And if you look at those pictures of the wheat there, you can see that they're all different, and they've all been introduced during that 100-year period. There's also the fact that nitrogen fertilizer and pesticides and fungicides all came in the sort of 1950s, 60s. So some of that acceleration you see there. So if you think this is winter wheat, we started out at 2 tons per hectare, and now we're up around 9, probably higher, depending on the farm, depending on the field. So, you know, we've seen a good increase in wheat yields. I wanted to say something that we did at NIAB, actually, which was we looked at diversity from the progenitor species of wheat. So if you look at Aegilops speltoides in Triticum muratu, so a chance hybridization brought those 2 species together 100,000 years ago. And that gave you wild Emma, which some of you will know. And 10,000 years ago, there was another chance hybridization with that wild Emma, with wild goat grass. You can cross this, what they call the AB genome, so the Triticum muratu and the Aegilops, with these other species, but the wild goat grass is less diverse. So you mostly see, if you look at the DNA, the wild goat grass is not variable. So what we did at NIAB, we resynthesized wheat using Triticum durum, which grows quite well in the UK, and with Aegilops tauschii. Now this all a lot of it had to be done in the laboratory. It had to be rescued, and the little red dots you see on the map there, that was where the collection came from. So we introduced a lot of diversity for the D genome in wheat. And again, this takes a long time. It's what I think a lot of people don't really appreciate about plant breeding. So even if, when you've done all of this, you still need 7 to 10 more years to breed new varieties from them. So durum wheat, Aegilops tauschii, this gave us increases in drought tolerance, better nitrogen use efficiency, better disease resistance, and higher yields. But not sufficient, not enough. You know, there was an improvement, but not good enough to actually feed the world in 2050. So thinking about molecular biology, and I'm sure most of you know this, I'm not a specialist in any of this, by the way, but you know that DNA, every cell in a living organism contains a copy of the same DNA molecule, and that DNA is transcribed by RNA and then translated into proteins. So differences in DNA, mutations. We went through this big debate, should we use the word mutation? People find it scary, but actually I can't think of a better word. So when you have a difference in a DNA, that's a mutation. And these differences are associated with fitness and disease resistance and genetic diversity. And that gives you a different protein. A Canadian farmer gave me this picture a while ago. And it is quite, if you think about it, it is quite interesting how the scientist and the businessman are trying to open up the GM box, coffin, and other people are trying to nail it down. And it reminds me of when I first came back to the UK, I was working with a group of people from Durham University, and we wanted to create nematode resistant, so the little worms in the soil, nematode resistant potatoes. And they got a protein from peas, which we put as much protein into a potato about this size as you would get in 5 peas, and you eat potatoes and peas together. And it was more resistant to nematodes, and the reason was that the chemical that was being used was about to be banned. It had already been banned in Holland. And it was at that point that the Soil Association and others in the organic movement decided you can't be organic and GM. Now, I didn't expect that to happen because I thought we were doing good for farmers, but that has continued to be controversial, even into the precision breeding. So this is really a simple way of looking at it, but if you think about genetic modification, if you think you've got a blue plant and a green plant, and they're completely different and they don't cross between each other, you can take a piece of DNA from the blue plant and put it into the green plant, and that will express a new trait. In genome editing, you just make one change in the DNA. And people talk about transformation. So transformation is the word that we use to describe the process by which we introduce new genes into plants and introduce the tools for gene edits. And it requires growing the seed in the laboratory in agar. And this is wheat. That's on the left-hand side. That's an embryo of wheat. It becomes a callus, and then you see little wheat plants coming out of it. They are perfectly normal when they develop. And this is Agrobacterium tumefaciens, which forms crown galls on some plants. It was taken into the laboratory. They removed the genes which cause it to cause a crown gall, and you can introduce the genes which you want to put in. So it's like nature doing it for you. There are other ways, but this is the way that most people use. And just to say that sweet potato, DNA sequences from Agrobacterium have been found in many sweet potato species, samples. And I don't know whether that's just sweet potato, but it certainly is in sweet potato. So it's not as if this is a completely new phenomenon. You know, Agrobacterium has been doing this for some time. So there's a lot of words on this slide. So there was a chap called Graham Brooks of PG Economics in the UK. He looked at research over the 24-year period, 96 to 2020, and he estimated that GM crop adoption has reduced carbon emissions arising from reduced fuel use, because insect-resistant plants, you don't need to go in and spray them, so you're not running your tractor over them all the time. And that was equivalent to removing 25.9 million cars from the roads. So this is his data. He also estimated that GM crops do reduce insecticide use. So GM insect resistance has increased yields in maize and cotton and also in soybean. And so that's made one of his comparisons is that the reduction in insecticide insecticide and pesticide is equal to 1.5 times China's total annual crop protection product use. So it's just some examples that really make you think. There's also the reduced impact on land use. So GM crops allow farmers to grow more without needing to use additional land because they are higher yielding. And he estimated that if crop biotechnology had not been available in 2020, maintaining global production levels that year would have required the planting of 23.4 million hectares, equivalent to the combined agricultural area of the Philippines and Vietnam. Now, how he got those numbers, I don't know, but that's what you'll find in his work. So when I was at NIAB, we came together with Cambridge University and we created something called the Crop Science Centre, because Cambridge works on very long-term research, and they say, well, maybe in 25 years we'll have an impact on something. And they really hadn't done anything in agriculture. So Professor Giles Oldroyd, who now leads the Crop Science Centre, also leads what's called the ENSA programme, funded by Bill and Melinda Gates. And his objective aims to reduce dependence on inorganic nitrogen fertilisers, which, it's a very interesting topic because the plant breeders are breeding for a system with high nitrogen fertilisers. So they might argue that they're not doing this, but actually that is what they're doing because the testing systems require high nitrogen fertiliser. And also he's working on reducing phosphorus application by increasing mycorrhizae, the small fungi that associate with roots. And all of these things need to be GM because wheat and cereals don't have bacteria that associate with their roots to fix nitrogen, whereas peas and beans and clover do. So it's great to have peas and beans and clover in your rotation, but it's not the same as having something which fixes nitrogen. And of course, the reason Bill and Melinda Gates are interested in it is that in Africa they don't have access to, or don't have access, they don't have the money to spend on these fertilizers. So that's a big motivator for them. So potatoes has always been my favourite crop, and I just wanted to say a few words about that I learnt very recently actually. There's a company called Simplot in the US. They're a major US food and feed company. So they don't just do this, but this is one of the things they do. So they have a biotechnology division improving potatoes and strawberries at the moment. In the US, there's 1 million acres of potato, 90 million acres of corn, 88 million of soybean, and 2.2 million acres of canola, which is big. But potatoes are quite small. But there are some big challenges with potatoes. They are perishable, so they're 80% water. So if we're not growing potatoes in this country, we don't want to think about importing them. Because, can you imagine? Just lorry loads of potatoes and coming across. Storage, providing potatoes year round. When they're stored, the sugar content increases and that makes them brown when they're made into chips or crisps. They bruise and they have big disease problems in the field. So what they have done is to use genes from wild and cultivated potatoes, no foreign genes, and a lot of this work was actually done at the John Innes Centre in Norwich because they've not been able to commercialise it in the UK, so they have taken it to America. And I just wanted to highlight lower acrylamide potential. So acrylamide is a cancer-causing agent which is generated when things are fried. So these potatoes have lower acrylamide potential and they also have late blight protection. Now, we can talk about late blight and how you do get some organic potatoes which grow quickly and grow slowly, and that therefore kind of escape the late blight. But it's not the same as when you buy Maris Piper in the shop or your crisps or your chips. They are often sprayed 9 times, 10 times, sometimes 15 times with a pesticide to control blight. And sometimes, if you're a farmer, it's like, is there going to be potato blight? Well, I think I'll just go and spray anyway. And I know they do that, and that's because they really need to protect their crop. So the market acceptance, they're in their 10th crop year, and they've sold over 1.4 billion servings in the US, mainly 70% food service and the rest retail. And they have a 1-800 number on the pack. 25% were the wrong number, which always makes me laugh. 25% asking where you buy the product, and you can see the other areas that people have asked about. And I think it is a big thing in the US to have labelling that people can relate to and people can go back to. So let's think a bit about mutations. So as I said before, mutations can arise naturally, but they're often the result of treatment with chemicals or irradiation. And we now have the tools to validate those changes and identify the mutations, but we still have to cross out all of the other mutations that we don't want. So how can we accelerate the process? And the answer to that is gene editing. These are corn cobs, and they're all the result of mutations in one form or another. But we've selected mutations. This is probably my favourite thing. You know, your Brassica oleracea, it's cabbage, it's Brussels sprouts, it's broccoli, it's cauliflower, it's Chinese cabbage. And we have selected the varieties that we want and they're all mutations that we want to use in our kitchens. So with gene editing, you can introduce the mutation directly without having to make a cross. So that mimics mutation breeding, but you don't use the chemical mutations or the radiation. And it's also precise. It just cuts that one place. And it's a technology called CRISPR-Cas9. And these are the two people who invented it. And just in case you think it might be something weird, it's already in your yogurt and your cheese. So there's quite it's quite a lot around. So how it works is this: a DNA binding protein is guided to a specific site in the DNA and then cuts it. The cell tries to repair the cut. It can't, or it usually doesn't. And then you get a mutation. Importantly, the DNA binding protein can be selected away in the next generation, just leaving the altered plant DNA and no inserted foreign DNA. And gene editing has been used in a wide variety of applications so far. It is, there's very little on the market. There's nothing on the market in the UK, but we'll come on to that. But there are things which are starting to reach the market in the US and in Canada. And one of the important things that people are very interested in is low gluten wheat. The polyphenol oxidase gene in potatoes and apples, you can turn that off with gene editing to reduce post-harvest bruising and reducing the application of pesticides, which is something that we really want to do. There are other examples in oranges and grapefruit, as well as in rice and wheat and tomatoes with mildew. I worked for years on pod shatter resistance in oilseed rape. And there is a gene which, when it's turned off, the pods don't shatter when they're dry. And that gives you not just better yield, but less weeds of oilseed rape. And there's, I think on the market in the US now, there's healthier oil profiles from soybean. And of course, we're very interested in the stress and climate resilience that come from those gene edits. So this is quite an old slide from 2020, talking about how different countries regulate. So if you look at the green, these genome-edited crops are not regulated as GMOs. They obviously are regulated in different ways. In Canada, they're not regulated unless the trait is identified as novel. In the US, most are not. It's a similar argument. In the European Union, we do get an odd perspective because of what the European Union's doing. Genome-edited crops are regulated as GMOs. And in the UK, there was a discussion ongoing post-Brexit. The government were like, now, what can we do to prove that Brexit was a good thing? Well, this was one of the things that they chose to do, which was to accelerate the landscape around genome-edited crops. So these countries consider genome editing as undistinguishable from traditional breeding. And most of them still have a notification process, so you still have to notify. And in the UK, the plant breeders have said they will keep a list of the gene-edited crops which are on the market. And so the new precision breeding bill removes genome editing from the scope of the GMO legislation to be aligned with other countries. So the EU ruled that genome-edited crops should follow the same regulatory process as genetically modified organisms, but that's being questioned now. So they're saying, actually, is it that different to what's mutation breeding? So the new legislation changes the definition of a genetically modified organism so that they're still GMOs, but the gene edits are different. It only deals with plants at the moment. And really importantly, it deals with the properties of the crop, not the process by which it was made. So that were, I don't think there's any other example of something which was regulated, even before there was anything on the market by the EU, where the process, that transformation process that I talked about, if you've gone through that process, that makes it a GMO. So in the UK, there's a new notification scheme for research and marketing, which I don't really understand, but that's true. There's no compromise on food safety. The Food Standards Agency has statutory responsibility to regulate food and feed. And the government believe this will attract inward investment to the UK and reduce impacts on the environment and greenhouse gases. And so they just want to accelerate this process. So the Precision Breeding Bill received royal assent in March 2023. But there are 38 statutory instruments which need to be put in place before it can become law. And until they're in place, those products are still classed as GM. Research trials have been made easier, which means that there's a notification process. It's not quite as onerous as the GM. The EU has draft legislation in discussion. Scotland, I know, and Wales, I'm not sure, want to remain in step with the European Union. Scotland because they think they're going to be independent and then they're going to come back into the EU and they want to have the rules the same. We hope that these statutory instruments will be in place before the election, because it's been going on for almost a year now. And the Food Standards Agency have examined this bill. In fact, in the beginning, even though the whole of Parliament said, we've got this new bill and we're all voting for it and it's all positive, the Food Standards Agency questioned it and said, hold on a minute, we're not sure about this. So they are really sort of independent of the government. So they conducted consumer research on this, showing that the more people know, the more they're willing to try. And they said that more than 60% would buy it if it was cheaper, more nutritious, less pesticides, but they did want labelling. So these labels are from the US. So in the US, you have either a stamp that says bioengineered food, which is an odd word for me, or a high likelihood of a GMO ingredient. But these are just for GM. I think most companies will also label gene-edited crops. So that's where we are with gene editing. And I also wanted to say that it's a time thing as well. So if you look at, this is GM product development, and if you just look at the time that it takes, and this is in the US, it takes, they're saying 15 years to get a GM product on the market. They're also saying that with gene editing, it's much quicker. Now, it might be different in the UK because we have this plant variety rights legislation, which they don't have in the US, but it's urgent. We need to do this, and I really believe that we have the correct regulations in place to make sure that nothing slips through the net, although I don't think anything has slipped through the net in the US. But we have the regulations in place, and I think that we will soon be seeing gene-edited crops in the UK. And farmers need it. I work part-time now for a farming company that grows lots of salads, and they are desperate for resistant plants. So this is a way to get it to them quickly. So just wanted to go back to NIAB and finish with this. So on the 14th of October 21, King George V and Queen Mary came to officially open the new institute in Cambridge. And the chap laughing in the corner there is Lawrence Weaver. He was the first director of NIAB. And there is Queen Mary planting this tree. So in 2019, which was NIAB's centenary, that's me on the right, we decided to reproduce this with our Queen. And we were told by Buckingham Palace that there is no way she'll plant a tree. You should give the spade to someone else. And so we kept saying this, you won't need to plant a tree. When she got to the tree, she handed her hand back to an equerry, grabbed the spade and said, I'm still perfectly capable of planting a tree. And it was all over the papers the next day. So that was a really, really great event. And yeah, I really did enjoy my time at NIAB. And so that's it really. I mean, I'd love to answer questions. I'm not sure I'll be able to answer all of them, but I would be really glad to do that.
Thank you, T. That was really, really interesting. So we've got time for questions. I've got a microphone. Yeah, I've got a question here at the front.
I don't need the mic. By sheer coincidence, this morning I was lecturing to my first years about Kathy Martin's anti-cyanin expressing transgenic tomatoes. She said a very interesting thing. She said in Canada, they regulate the trade, not the technology.
That's correct, yeah. Yeah. Yes, I mean, I worked in Canada for 5 years, and it was like that then. And at first, they captured all of plant breeding. So they said any novel trait goes through this system. And plant breeders were up in arms because, it's a new disease resistance that they've introduced, and they didn't want it to go through these regulations. So I think that was sort of quietened down a bit. But it is true, they regulate the trait, not the process, which is I think it's only in Europe. Yeah, the opposite. It's the process that they operate. And those tomatoes, they are amazing. They've got them into the home market, so for people to grow them in their gardens, and they're flying off the shelves. They've got different anthocyanins in them. So people say, oh, well, you can get purple tomatoes, yes, but not with the same level of these anthocyanins as Kathy has in hers. They are really amazing.
Thank you. I've got another question here. Thank you.
Thank you. Is it working?
Yeah.
Good. One of your early slides, you showed a between 60% and 90% increase in food demand by mid-century. And I was wondering about the assumptions that was based on. Is it based on the current balance between crops and animals being maintained at that time? And if it was, or if it wasn't, if you were to decrease animal numbers and the amounts of cereal and soya which is fed to animals, how much difference would that make to the percentage increase that you anticipated?
That's a very good point, and I don't know is the answer. But yeah, it is a good point, because most of the maize, most of the wheat, most of the barley is fed to animals. So if you reduce the number of animals, you wouldn't need the same amount of crops. But I imagine it's based on the status quo. I mean, it must be really difficult to project otherwise.
Because as a devil's advocate, you could say that you'd gain much more by changing those ratios than you're likely to do in the time scale by the breeding, and more economically.
Yes, but that's for those major crops, isn't it? But there are obviously reasons why we need to also protect the minor crops, because they don't have the chemicals they don't have the protection that we need, and so we have to find ways to do that too.
Thank you. Any other questions? Yes.
Just thinking about insects and birds and things. What implications might GM have on them?
Well, it's interesting, isn't it? Because neonicotinoids were banned because they were seed treatment for oilseed rape. They were banned because they persisted into the flowers, and there were experiments that showed that affects bees. And there are various views on this. I mean, there are people who say, well, most bees use oilseed rape as a source of honey. At least they did in our area. So, there's a sort of a balance there. So with the genetically modified crops, that is all tested before they get onto the market. And I don't know of any examples where insects have been a problem. I do know an example of, I think it's the insect-resistant cotton. So in India, they don't spray now with insecticide, so they now have another problem with aphids, with other insects. So there can be knock-on effects like that. And of course, there's the whole glyphosate story that I thought somebody might bring up, which you have plants. There were soybean and maize, and they were all resistant to glyphosate. And farmers were like, whoop-de-doo, we'll just grow maize after soybean after maize after soy. And of course, they got weeds. But that wasn't due to GM. It was due to the way it was used and them not being advised properly in the rotation.
Thank you. Another question up here.
Thanks very much. That's a really interesting presentation. Really, lots of facts in there. How can you help us do two things, really? Persuade Welsh Government that gene editing is not gene modification. And secondly, can you help us adopt the precision breeding bill in Wales, as you have in England? Because, we desperately got really good plant breeders here. They can embrace the technology, and yet our government is not willing to embrace it at the moment. So any help we can be, please tell us.
I think that genetic modification and gene editing are just two different things. Genetic modification, you're taking a protein from a different species and you're putting it in. With gene editing, you're usually knocking something out just by changing one base pair. So there is a difference. The only commonality is in the EU. The process by which you introduce those tools into a plant is the same. So that's why they're regulated the same. And I don't know how you persuade people. I mean, we did have lots of discussions with NIAB was the founder member of something called the All-Party Parliamentary Group on Science and Technology and Agriculture. And of course, that's across all parties. And it was quite difficult to get the Labour Party and the Conservatives to agree, because they're just politicians, They don't care whether it's right or wrong. They just set themselves up against each other. And that's basically what happens in the EU. So I don't know, other than by talking to them and trying to explain.
Thank you. Any more questions? Yes. I've got two here.
I imagine these crops are going to cost more money to produce, and the sales will be higher. Are the farmers going to be able to afford them?
Well, I would say that if they are not using the insecticides, those things cost a lot of money. The pesticides cost the farmers a lot of money. And if they can get plants that fix nitrogen, then that would be, the farmers will save money, I think. And I just think it's a technology that will be coming, and plant breeders are going to have to use it because it's so much more efficient than what they're currently doing.
Will the manufacturers have a monopoly on it?
Well, that's what happened with genetic modification, wasn't it? I mean, when I first started doing it many years ago, in the 90s, we thought that all we were doing was introducing new variation into a plant breeder's toolbox. But basically, the chemical companies decided that they were going to make sure that genetic modification was regulated in exactly the same way as their chemicals were regulated. So if you look at they are almost the same. And that meant that no one else can afford it except for them. That's my view anyway.
Thank you.
Yeah.
I was interested in the evidence you showed about public acceptance of gene-edited crops. Do you think we need stronger links between the nutritionists, food scientists, and crop breeders? I mean, it must have struck us all about the incredible number of column inches on one diet fad or another. Every week in the paper there's a different one promoted, let alone the millions that must be made on social media adverts for all of this. Clearly that's because there seems to be a public thirst to assuage our guilt about our diet or improve our health and well-being, and we chase after any sort of fad with that. But actually getting a scientific basis for our raw ingredients to be more nutritious and more healthy, if genuinely there is an evidence base for that. How key do you think that is to improving acceptance?
It's true, again, of this farming company, G's, that I work for part-time. They are looking for radishes which are less sharp because young people apparently don't like that flavour. But they want to, lettuces with a higher nutritional content. They're just sort of surveying what's out there, and they as farmers want to do that because they want to introduce to the supermarkets these new products. But yes, you're right. There ought to be more contact between nutritionists and plant breeders. I'm sure there are in some companies, like when I worked for Limagram, which was a seed company, a French farmers cooperative. They had a lab. It was mainly for wheat, but they had a lab that was looking at different nutritional properties of wheat. So some of them do. But in the end, the variety that goes on the recommended list is the one that's the higher yielding and, with all the chemicals that you can throw at it.
Thank you. Any more questions?
This topic is the reason why I'm really interested in going into plant breeding, because it's a serious debate in my country. If we should go to the market with a healthy looking corn, with GM, and a traditionally grown corn that's not looking healthy, most people will go for the one that was produced traditionally. Why? Because of the fear of chemicals. Currently in Nigeria, we are having issues with weevils because when you plant your beans, you have to use fungicide to protect them. During storage as well, you have to use fungicide, and that has led to food poisoning. So farmers, researchers, everyone is looking for what do we do to have good beans without fungicide. While you were giving your speech, and that was the only thing I wrote down, I was like, should I ask? Shouldn't I ask? But I think I should ask. You mentioned resistance to nematode using protein from beans. However, the chemical has been banned. So I wrote here, chemical mutagens. Could that be the reason why these farmers, researchers are being scared of GM-produced food because of the chemicals?
Well, I think one of the reasons that I don't know about Nigeria, but I know in some other African countries, they were looking to the EU and they were saying, well, there's obviously a problem, so we're not going to import. So there were even examples where, people were starving, I guess, and the US was sending maize over and it was GM, and so they didn't want it. So yeah, I think there's a lot that can be done to avoid chemical use. It's the process is much more difficult with peas and beans, but it can be done.
Thank you.
Thank you. Well, I'm not seeing any more hands raised. We have got refreshments outside, so we can continue the discussion and the conversation afterwards. Could I ask everyone to give a big round of applause once again for Gina? Such a great.
Thank you.